ఇంటర్మీడియట్ విద్యారంగంలో అగ్రగామి మన NSR ఇంపల్స్ ఎడ్యుకేషన్ ఇన్స్టిట్యూషన్. ఐఐటి జేఈఈ అయినా నీట్ అయినా తిరుగులేని విద్యా సంస్థ NSR ఇంపల్స్.
Hello students, this is NSR Impulse Junior College and here I am going to introduce about the concept called morphology. Now basically in biology we have different branches are there for us to study. In your intermediate we have biology is being divided into the two main branches, one is botany, other one is zoology. So here I am going to discuss one of the branch of biology, that is botany. So as I told you, biology has two important branches, one is botany, other one is zoology. So here this is Venkateshwari, senior botany lecturer, who is going to take up the subject botany here. And in that let's going to start with the basic part of the botany because in NEET medical entrance exam, biology and physics chemistry has lot many concepts, but they are very easy if you can follow and if you can understand, because all expert team will deal with you in bringing hard topics into simpler topics. So everything is going to become very easy if you concentrate and if you study well. So welcome to the first year junior intermediate classes. So let us start with botany class. So here botany is the one which we are going to start with. It's nothing but one of the branch of life science. In botany mainly we are going to discuss about a different type of plants and different type of what you call prokaryotic cell, eukaryotic cell, differences between a plant cell and animal cell. So I am not going to take you to the big part, and what I am going to speak now are nothing but the basics. Few concepts are being studied in your 10th class. In 10th class you have studied very clearly a difference between a plant cell and animal cell. In 10th class you studied very clearly the differences between a plant cell and animal cell. First of all, why do we have to divide biology into botany and zoology? Here zoology is mostly we deal about animals. Animals, we are nothing but human beings, we are also animals. So here the related to animals you are going to study in the branch called as zoology. Now related to animals, related to human beings, and also we are going to study about plant cell, this constitutes mostly botany. I will tell you the simple thing, what is the importance of botany to study in biology as one of the major criteria along with zoology. You know that today even if you crack the good rank in NEET, you will become good doctors, right? You will complete your medicine. And you know, for all our human beings, basically in medicine we always talk about health of the human beings. When you talk about health of the human beings, of course you study the human anatomy and you study what are the diseases which are going to collapse the human health and all that. Now in that mainly the major part comes, you need to give solution to the patient whoever come with a different sort of diseases. So at that time the solution may be in terms of what is called medicine. The solution may be in terms of medicine. So where do we get these medicines to treat a different type of diseases in humans? We get 98% of the medicines are alone which we get from the source called plants. 98% of the medicines which mainly we get from the source called as plants, and only 2% of the medicines we get from other organisms called microorganisms. 2% of the medicines today in generally, if you all have to understand in terms of antibiotics. So we know penicillin, streptomycin, erythromycin, azithromycin, different type of antibiotics, painkillers, and general terminologies generally we call medicines. Whatever we use the medicines to treat the health of the human beings, in that 90%, 98% of the medicines which we get from the source called plants, the plants for our understanding which we call them medicinal plants. Okay, so basically if you talk about the plant, even you need to know which part of the plant, which are the medicinal plants, where do you find them growing, and which part of the plant is very much useful in the preparation of medicine, and what are the chemicals present in the plant. Pharmaceutical people will be in continuous search of identifying a new medicines, new chemical extraction from different type of medicinal plants, right. Sometimes the leaf nicotine, or ashwagandha, raw wolfia, ashwagandha is nothing but most familiarly you might have heard about the example ashwagandha, most familiar one. In ashwagandha root, this is called as king of medicinal plant. Why? In that root is very much useful, shoot is useful, leaf is useful, flower, inflorescence, all the parts of ashwagandha are very useful in the preparation and the formulation of different type of medicines. Okay, so mainly we are going to talk about the things like genes, genetics, biotechnology, molecular biology. These all are very interesting topics. To what extent we go means, we go to the level of cell. Inside the cell, nucleus we talk about. Inside the nucleus we talk about the DNA, RNA, genes, and what extent the genes are responsible for expression of genetic characters, and how they are inheritable, means how they get transferred from one generation to another generation, what not, lot many interesting facts, most interesting concepts we are going to study in botany along with zoology. So, botany and zoology, some of the topics will be very common. So, continuation of one part we will study in botany, and continuation of that other part we will study in zoology, right. So, here the main use of studying the botany here is we are going to study different type of medicinal plants, and the medicines are being extracted from different parts. Which are those parts, and what is the chemical composition of different type of plants. Like some of the plants are very much rich in alkaloids, flavonoids, terpenoids, right. So, we know pharmaceutical people, after doing a lot of research, they will come to know that okay, diterpenoids have lot of medicinal value. So, we go for chemical analysis of the plant. How do we do the chemical analysis? That is also big topic, right. It has lot many different methodologies and different type of techniques to extract the chemicals from the plant. Each chemical has its own chemical value, right. So, suppose one research come and say that okay, wherever you find diterpenoids, they have lot of medicinal value. Then automatically we look for the plant which has more diterpenoids. What are terpenoids? A type of chemicals. Where do you find them? In some of the plants. They are nothing but natural chemicals. But these natural chemicals have vulnerable ability to kill pathogens. Pathogens means bacteria, virus, fungus, right, are different infection causing organisms. So, these different infection causing organisms can be treated properly with the extraction of a proper and correct type of medicine directly from different sources of different types of medicinal plants, right. So, here when you talk about this part, this is how we start with the botany. Botany is very important. So, to support the intermediate syllabus, already in your lower classes you studied very clearly the difference between plant cell and animal cell. So, first of all, if you want to start with the botany, guys, all of you know it very well, cell, this is nothing but structural and functional unit of life. Okay. So, what we talk about here, first of all, what is a cell? Cell is nothing but structural and functional unit of life. So, if we talk about structure and functional unit of life, what exactly meaning of that? In chemistry you might have studied about atoms, molecules, all molecules join together. The lowest molecule, which is very basic for the life, is nothing but what we call cell. What is the basis for life? Cell. You know, in the course of evolution, in the very beginning of earth formation, in ancient period of time, you know that on earth we find only unicellular organisms. You know, in generally the organisms are divided into two categories. So I am talking about living organisms, unicellular organisms, and multicellular organisms. So, in the very beginning of the formation of the earth, in ancient period of time, in the course of evolution, if you look at, living organisms are in generally classified into two categories: unicellular organisms and multicellular organisms. You know, unicellular organisms means they have a single cell, only one cell is there, and you know, you can take best example, what not. You are very much familiar with the example called amoeba, yeast, right. So what are there so many unicellular organisms, like even microscopic organisms if you take, most familiar one, bacteria, right. So our life is being evolved with single cell. A life is being evolved with single cell. And here these unicellular organisms used to colonize in the environment. Colonize meaning they make different colonies, they group together and they used to survive in the very beginning of, in the very early days of formation of life on earth. Then slowly the unicellular organisms undergone many modifications due to changes in the environment, and that lead to, due to changes in the environment, mainly that slowly lead to formation of multicellular organisms. What I am trying to tell you here, unicellular organisms undergone many modifications and many changes due to changes in the environment, due to impact of environment. Unicellular organisms now they slowly developed into multicellular organisms. Now these multicellular organisms later, for our understanding, we again classified them into two categories. What are they? Prokaryotic cells and then eukaryotic cells. Okay, what we are studying? Prokaryotic cells and eukaryotic cells. Then what are these prokaryotic cells? Now if you look at these prokaryotic cells, now we try to differentiate them very clearly here. We will try to differentiate between the prokaryotic cell and eukaryotic cell. Probably in your lower classes you studied very clearly. Now if you look at the cell, we know that membrane bound cell organelles, are absent in case of prokaryotes, and these membrane bound cell organelles are present in case of eukaryotic cell. Membrane bound cell organelles are absent in prokaryotic cell and membrane bound cell organelles are present in case of eukaryotic cell. So what exactly meant by membrane bound cell organelles? So probably in your lower classes you studied well maybe the plant cell and animal cell. You know, cell is made up of a different type of cell organelles. The cell is made up of different type of cell organelles. What are the different type of cell organelles? So, you know the cell organelles like mitochondria, chloroplast, nucleus, right, what not, Golgi complex. So, these all are nothing but we call cell organelles. These all are nothing but what we call cell organelles. You know that they are surrounded by the membrane. That's why we call membrane bound. What exactly meant by membrane bound? They are surrounded by membrane. That's why they are called membrane bound cell organelles. Membrane bound cell organelles. I think you know, you studied about structure of mitochondria, structure of chloroplast, structure of nucleus, right, and even studied about Golgi complex. Now these all cell organelles we can see very clearly in well developed organisms like a plant cell and an animal cell, but not in the unicellular organisms, first evolved organisms like prokaryotic cell. Why? Because prokaryotic cells are unicellular organisms. Prokaryotic cells are unicellular organisms. They have only single cell. So, what happens there? All these membrane bound cell organelles are totally absent, but they are present in case of eukaryotic cell. Why? Because eukaryotic cells are multicellular. Prokaryotic cells are unicellular and eukaryotic cells are multicellular. Now here membrane bound cell organelles are absent in prokaryotic cell and membrane bound cell organelles are present in case of eukaryotic cells. So in case of prokaryotes we find primitive nucleus is present. Primitive nucleus, it's called nucleoid. It's present. What exactly meaning of primitive nucleus, nucleoid is present? That means here I say membrane bound nucleus is absent. Membrane bound nucleus is absent. Look at the things which are absent. Then you get a question straight away. If I write this first as membrane bound nucleus is absent, then definitely in your young mind definitely you will get a question or doubt. That means prokaryotic cell is lack of nucleus. That means nucleus is totally absent in prokaryotic cell. No, nucleus is present but nucleus is not surrounded by membrane. That means membrane bound nucleus is absent. Membrane nucleus is not with membrane. Nucleus is without membrane, but nucleus is present. What is that nucleus? That is a nucleus is the first formed nucleus observed in first formed organisms on earth. What are first formed organisms on earth? Unicellular organisms. What do we call them? They are called prokaryotic cells. Does you think in prokaryotic cells are lack of nucleus? No. Prokaryotic cells also contain the master of the cell. Who is the master of the cell? Nucleus. Okay, but that is first formed nucleus. First evolved nucleus. What is that first evolved nucleus? Primitive nucleus. This primitive nucleus, first formed nucleus in first formed organisms like unicellular organisms, it's called nucleoid. What's the meaning of nucleoid? Nucleus without membrane. What is nucleoid? Nucleus without membrane. That's why it's called nucleoid. Nucleoid is present but membrane bound nucleus is absent. There is a difference. So in coming classes you will come to know the difference between very clearly what is nucleoid and what is membrane bound nucleus. Presently you try to understand in prokaryotic cells nucleus is present, that is primitive nucleus, that is first formed nucleus and it's called nucleoid. So, nucleoid is present, but double membrane bound nucleus is absent, meaning membrane bound nucleus is absent. There is no clear distinct nucleus, there is no clear distinct membrane. Right, coming to eukaryotic cell, here membrane bound nucleus is present. Membrane bound nucleus is present. Alright, so here these are the very important differences here. Right, now apart from this, of course there is cell organelles are absent, we said membrane bound cell organelles are absent, membrane bound cell organelles are present here. Here membrane bound nucleus is absent, but here membrane bound nucleus is present. So, these are nothing but prokaryotic organisms, first formed unicellular organisms. They lack of very important components. They are first evolved organisms on earth, and later there is a lot many changes and modifications and lead to formation of multicellular organisms, or we say multicellular organisms are evolved from unicellular organisms. And to differentiate, suppose I have given one cell from multicellular organism, I took one cell, and from unicellular organism I took the same cell. When I place it under microscope, what are the major differences? To identify it is a prokaryotic cell and this is a eukaryotic cell. Prokaryotic cell does not have membrane bound cell organelles. What are membrane bound cell organelles? We say mitochondria, chloroplast, nucleus, Golgi complex are absent, whereas they all are well visible in case of eukaryotic organisms. Similarly, now if you look at, nucleus is the master of the cell. Nucleus controls all the functions of the cell, right? That is, membrane bound nucleus is absent in prokaryotes. Membrane bound nucleus is present in case of eukaryotes. Then you will get a small question over here, that means nucleus is totally absent in prokaryotes. If this is a question, if you get in your young mind, definitely the answer is no. Why? Nucleus is present, but that is without membrane. Then what do you call that? That is primitive nucleus, meaning first formed nucleus. It's called nucleoid. What's the meaning of nucleoid? Nucleus without membrane, it's called nucleoid. Nucleus is not totally absent, it is present but no membrane. It is present without membrane, it's called nucleoid. Other differences are there, but when we go with the syllabus we will discuss in detail. Presently I am trying to give you introduction about what are uni, what are multicellular, what are prokaryotes and what are eukaryotes. Now these eukaryotic organisms are further divided into, we say, so when you say this is prokaryotic cell or the organism, similarly eukaryotic cell, one cell if you take it, that is from eukaryotic organism. Now these eukaryotic organisms are very clearly differentiated into two categories. What are they? Plants and animals. What are they? Eukaryotic organisms are further very clearly differentiated into plants and animals. Now it's very easy for you now to differentiate plants with the animals. Now this is where the clearly you need to talk about a structure of the plant cell and animal cell. Now if you observe the plant cell, you know that plant cells are characteristic of containing cell wall. Plant cell is characteristic of containing cell wall. What do I write now here? Cell wall is present. What do I say? Plants, cell wall is present. Coming to animals, what we write? Cell wall is absent. Cell wall is present and cell wall is absent. Now you can make out a clear difference. Suppose suddenly I call you to the lab, I have given the microscope, because cell is visible only under microscope. Now under microscope I have placed 2 microscopes and with the complete focus on with on cell in one microscope, other microscope is also focused with a cell. Then how do you differentiate plant and animal cell? I said, guys, maybe this is one microscope having plant cell, one microscope having an animal cell. Find out which microscope has a plant cell and which microscope has a animal cell. Then the first thing what you need to identify, the first thing what you need to look for is cell wall. If you look at on the boundary of the cell, if the cell wall is present, it's called a plant cell. If the cell wall is absent, definitely you can identify that as an animal cell. Right. Then apart from this, even you can take vacuole. A bigger vacuole is present and a big vacuole is absent. When vacuole is present, that definitely we call this as what you call a plant cell. When vacuole is absent, it called as a animal cell. Then what else can make us to differentiate? Apart from this, even we can say that chloroplast. Chloroplast is a pigment. Chlorophyll is a pigment. Chlorophyll present. Chlorophyll pigment is present in the cell organelle called chloroplast. Chloroplast is present and you know that makes the appearance of green color. So what we are doing here, we are making out clear differences between plant cell and animal cell. So what's the first thing you have to look for to identify the cell in the microscope? You need to look for cell wall. Cell wall is present or absent. If cell wall is present, then it is a plant cell. If the cell wall is absent, then it is a animal cell. The next parameter what we need to look for? Vacuole. If vacuole is present, then definitely that is a plant cell, and if the vacuole is absent, then that will be an animal cell. Apart from this, and the most another important difference is chloroplast. So they may be all simple points to differentiate plant and animal, but they are important. So wherever we keep star mark, those points are important to remember, even though they are very simple basics. Chloroplast is present because green parts of the plant contain chloroplast. You know, in case of animals, chloroplast is absent. Vacuole is present, absent. Chloroplast is present and it is absent. Then what is present in animal? If you look for, then there is a very important cell organelle called as centriole. Very important cell organelle called centriole. I will show you in the diagram, do not worry. The centriole is present in case of animals. Why do I keep 3 star marks here? Because this is the cell organelle present in animals and the centriole is absent in plant. So this is very important. This centriole is absent in case of plants. Right. So very importantly, you need to look for the last point. Centriole is a characteristic. Centriole is a cell organelle characteristic of present in case of animals, and centrioles are totally absent in case of plant cell. Then all of you might be thinking, what are the centrioles? Then let us discuss about centrioles, and I will show you not only centriole, I will show you the chloroplast, vacuole, cell wall, and let us make a clear difference. Let us revise once the basics. Clearly diagrammatic representation of plant and animals. Let us see what is present in plant and let us see what is present in animal. So here let us talk about plant cell. So if you look at plant cell, this is how we draw the plant cell. This is a plant cell. Right. So here Basically this is how we differentiate plant cell from the animal cell. So this is nothing but the plant cell. Plant cell contains, let us make similarities, let us mention together similarities in the center. Whatever differences are there, let us write on either side. Now what is the very first thing we have to look for? The boundary of the cell. So if the boundary contains the three walls, then this is nothing but characteristic feature of cell wall. This is nothing but the characteristic feature of cell wall. Cell wall, of course, it has three layers. Cell wall has three layers that we will study later. We call middle lamellum. This is middle lamellum, primary cell wall, secondary cell wall. That part we will study later very clearly. Now try to understand, cell wall is not the single unit layer, it is a combination of three layers. Cell wall has three layers, that is why it is clearly identified in the plant cell, and we can clearly distinguish that cell wall is absent in case of animal cell. That is the very first point. What is that? Cell wall is present, then you identify that cell as plant cell. If the cell wall is absent, that you will identify the cell as animal cell, right. Now, so here if you look at 90% volume of the cell, 90% volume of the cell, it's occupied by an empty space called vacuole. There is an empty space that's filled with some fluids, minerals, but this empty space, it's been bounded by a layer with empty space. It's called vacuole. So 90% volume of the plant cell, it's filled with a bigger one, it's called vacuole. With this we can clearly identify the plant cell from animal cell. So what is the second point we write? Vacuole is present, that is 90% of the volume of the plant cell is occupied by a bigger one called vacuole. Vacuole is present in plant cell and absent in case of animal cell. Then importantly if you look at, we have what's called chloroplast. So this is what I am drawing, chloroplast. This is how it looks like. It is a double membrane bound cell organelle. Inside we find thylakoids, groups of files of coins of arrangement of thylakoid. This is T I am writing. Thylakoid you might be studied in your lower classes. If not, we will discuss in detail in the first year syllabus, no need to worry. So if you can identify this, I am giving just boundary of the cell here. So here this particular boundary of the cell, what you can see structurally here, it's called chloroplast. So chloroplast is present in plant cell and chloroplast is absent in animal cell. Okay, so what are the three things we mentioned here? First one is cell wall. We can see very clearly the cell wall as an outermost boundary. It is not the single layer, have the combination of three layers. Together it's called cell wall. Inside the plant cell, 90% of the volume, 90% of volume of the cell is occupied by vacuole. Means it occupies bigger space, more space. So this is the second character to identify plant cell. And third character is what? Presence of chloroplast. Plant parts are green in color. Green in color, it's mainly because of chlorophyll pigment. Where the chlorophyll pigment is present? It's present in an important cell organelle called chloroplast. Right. So cell wall, vacuole, chloroplast are present where? They are present in the plant cell and they are absent in the animal cell. Right. Then what is the next one? Centriole. Let's come back to this. But before to this, inner to the cell wall, we find another layer within the plant cell. Itself it's called as, I am drawing the innermost layer, that is nothing but cell membrane, or itself is called plasma membrane. Okay, So inside what we find, it's called cell membrane or plasma membrane. So guys, let me ask you simple question. What is the outermost layer of plant cell? Then you need to choose the answer as cell wall. For example, in case of animal cell, outermost layer of animal cell, then you need to choose the cell membrane or plasma membrane. So animal cell is surrounded by a layer, that layer is called plasma membrane or cell membrane. This is common for both plant cell as well as animal cell. For plant cell also it is present, for animal cell also it is present. For both it is present. For plant cell it is present, for animal cell it is present. But what is the expected question here? For outermost layer of plant cell means answer is cell wall. Outermost layer of animal cell is cell membrane or plasma membrane. Second most layer of a plant cell is plasma membrane, that is the outermost layer of animal cell. See, look at the question, very simple question, right? It's a very basic question, right? So what is the answer? Outermost layer of plant cell means you have to look for option as cell wall. The same question asked about animal cell means you have to look for plasma membrane or cell membrane. Why? Because as we discussed the first point, cell wall is present in plants, absent in case of animals. Right. Now for both the cells, who is the master of the cell? Guys, what is that? Exactly, nucleus. So here we need to talk about nucleus. So this is the nucleus. This is the nucleus. So we find nucleus here. So how do you differentiate in plants? In plants, nucleus is pushed to one corner. In plants, nucleus is pushed to one corner. In case of animal, it is almost somewhere in the middle of the cell. That is double membrane bound. Two membranes are there. This is nothing but what you call nucleus. So I am writing here, nucleus is a common thing for both plant cell and animal cell. Right? Nucleus is common for both plant cell as well as for animal cell. Here also double membrane bound nucleus, there also double membrane bound nucleus. So here inside the nucleus what we find? So we find what is called chromosome, or we find DNA, chromosome. Inside the nucleus what we find? Chromosome. Chromosome at one corner, chromosome contains nucleic acids. So what are nucleic acids? Nucleic acids are nothing but DNA as well as RNA. What are nucleic acids? DNA as well as RNA. Inside the nucleus what you find? Chromosome. Chromosome contains what? Nucleic acids. What are the nucleic acids? DNA and RNA. So no need to worry, we are going to discuss each and everything once again clearly, because lot many points are there. We need to go little more depth of this, and you are going to learn so many new important aspects about DNA and RNA, because they are very important genetic material which gives unique characteristics for a living organism. So lot more interesting concept is pending. First of all, let's complete the step of having the basics. Maybe you know few basics here. Let us recollect it all and get ready for to have the lesson, right? So here nucleic acids are DNA as well as RNA. So here also what you find is nothing but the chromosome is present that contains same, and here and there you find a nucleolus. So here at one corner you find other material called nucleolus. So what is nucleolus, what is the chromosome, what is DNA, RNA, nucleus, in detail we are going to study later. First let us see what are present in plant and what are present in animals, right? Now in case of both plants and animals, they are surrounded by endoplasmic reticulum. All of you know it very well. Right. So here also in case of animal cell also endoplasmic reticulum is present. Right. So here endoplasmic reticulum is present in both the cases. So here we say this is endoplasmic reticulum. What is this? This is an endoplasmic reticulum. Now endoplasmic reticulum we have two types. What are they? Rough endoplasmic reticulum and then smooth endoplasmic reticulum. I hope you know clear differences between rough endoplasmic reticulum, smooth endoplasmic reticulum. What is rough endoplasmic reticulum? Rough endoplasmic reticulum is with ribosomes and smooth endoplasmic reticulum is without ribosomes. Without ribosomes. You know this difference well. So I'll draw here. This is nothing but ribosomes are here. So we call this rough endoplasmic reticulum. No ribosomes on the endoplasmic reticulum. This is called smooth endoplasmic reticulum. Now coming to this, what do we write here? Same. Here when endoplasmic reticulum contains ribosomes, it is called rough endoplasmic reticulum. Without ribosomes, it's called smooth endoplasmic reticulum.